Good morning, everyone. Maybe let me share my screen here first. Let me see here. Share. You guys can see that?
Yep. Yes.
Yes. I'm a senior scientist, senior staff scientist with Ocean Networks Canada. My background is on deep sea biology. And well, I thank Jasper and Romina for inviting me, for giving this brief presentation. I'll be talking a little bit about some projects we have at ONC, research projects combining video and acoustic imaging and passive acoustics for coastal and deep sea biodiversity monitoring. I want to acknowledge all my co-authors here, which have helped me to develop all these projects. So I guess I won't have to devolve too much on the review of the ONC's observatories, because Jasper did a great job describing the Neptune and the Venus observatories. I'll briefly mention some environmental changes that are happening in the Northeast Pacific. And of course, we are interested in using the video and acoustic data to monitor the communities with respect to those changes. In particular, I think the bread and butter of our video imagery is monitoring long-term change of benthic communities. But I'll also present some results of this, what we call this Fish Acoustics Experiment, some short-lived few-month deployment of this perturbation experiments, which we then combine video, acoustic imaging, and hydrophones to study animal behavior and a series of other aspects. And now, of course, since this workshop is on machine learning, as Jasper hinted, we are kind of starting to move towards machine learning tools, and I'll show you a few examples, some papers recently published, and we will have a next presentation that will give you a bit more detail on one of these machine learning projects. Well, this is a slide just to show what kind of processes ONC. This slide fits well with the Northeast Pacific. Of course, we have all these other monitoring locations in the Atlantic now, in the Arctic, but we're interested in biological, chemical, and physical processes and geological processes at various time scales and spatial scales. And here, of course, this is the Neptune and the Venus observatory that I'll be talking about. And Jasper, well, I mentioned and covered the way that one of our core mandates is to make all the data available. So you have multiple tools for exploring the data, downloading, and visualizing a signal before you even download the data. Here, for example, is a plotting utility. So we know that the Northeast Pacific is changing rapidly. There's long-term changes in temperature and declining dissolved oxygen. But I think what is more has been calling our attention recently has been the marine heat waves. For example, we had in 2013 that lasted for 2 years, more or less 2.5 years. The blob, which was composed of this this large patch of the Pacific that was almost 2,000 kilometers wide and affecting about the mixed layer depth, about 100 meters of water. And the water was warmer up to 4 degrees, warmer than the 30-year average. And of course, this had a pretty huge impact in the ecosystem. There was a substantially reduced phytoplankton and kelp productivity and biomass. There were shifts in the structure of zooplankton communities. We have those massive migratory bird die-offs, large mortality of marine mammals. So this hugely impact the ecosystems. And we had another marine heat wave a couple of years back, 2019, 2020. There was a 4-peaked sea surface temperature anomaly. And this study here, published by Chen et al. And Geophysical Research Lab, showed that this particular marine heat wave, they used a 70-year average of sea surface temperature data, and they found the oscillations of this, they call the blob index. And this last marine heat wave was significantly above the average for this anomaly index. So it's pretty dramatic what's happening. And of course, we want to monitor what the organisms and how this is affecting biodiversity. And as I mentioned, so I guess our bread and butter of our video imaging systems on the seafloor, in this particular case, we're in Barkley Canyon, we're monitoring long-term changes on the benthic community abundance or our community structure. And of course, we can study specific, the whole entire community, or we can focus on a single species. This particular study that's in review on deep sea research. So we're studying the pink urchin, this deep sea urchin that's, we used almost 7 years of video imagery. The Neptune Observatory, we are amounting to almost 10 years of video with some gaps here and there. We had some trawling heat events that took out our infrastructure for a couple of years. So we had some gaps, but in any case, this is the type of study that we support mostly. And we are able to track the changes of the abundance of these organisms with respect to this environmental change. And in this particular case, the urchin is particularly responding to the decline of dissolved oxygen and the temperature anomalies related to the marine heat wave. So we are able to, since we don't want to wait 7 years to obtain some really interesting, results, we want to study other aspects of the community. We want to study behavior of a specific species or some commercially important species. So with collaboration with Francis Juanes at UVic and his PhD student, Dan Xavier Mui, who's going to give a presentation to us later. So we devised this Fish Acoustics Experiment, which we then combine video, acoustic imaging, and a hydrophone in the same field of view. So we were able to, we actually used the bait. We wanted to maximize the detection of animals. So of course, we wanted to study species-specific behaviors. We wanted to compare the efficiency of video and acoustic imaging in this particularly turbid environment. So that this site at the Strait of Georgia, you see in the map above, is right off of the mouth of the Fraser River. So lots of sediment and suspension. So we thought that the acoustic imaging could be a cool device to help us monitor this species. We wanted to understand the role of noise and light disturbances, investigate soniferous species in the area. And of course, we still are able to do our regular inventory of the community structure in the area towards learning about the biodiversity. And of course, we want to, this is a lot of data as Jasper highlighted. So we wanted to, of course, apply machine learning tools to help us interpret and analyze this massive amount of data. So below here, you see some schematics of how we wanted to make sure that we overlap the maximum, the field of views of the sonar and the camera, and we put the hydrophone in the field of view of the camera as well. So this is how the seafloor looks like in a good day of visibility and a poor day of visibility. Right at this is above, you have a picture of the freshet when the sediments in suspension are at the highest concentrations in the area. So then we use the IRIS 3000, and you're going to see in the next few slides, we use another acoustic camera for the deeper site at Barkley Canyon. It's essentially a dual frequency sonar that uses two different detection and identification frequency, and uses 128 acoustic beams, and you have beams, sorry, the field of view 29 degrees wide by 14 degrees tall. and you, depending on the frequency used, you can have various ranges from 5 to 15 meters. So this is a schematic of our sampling protocol. Of course, the high frequency hydrophone was always on, and we turned the acoustic camera every hour for about 17 minutes, and then the video camera elapsed after 6 minutes elapsed from the acoustic camera. We turn the lights and the video on for 5 minutes, and then we turn it off, and we had another 6 minutes at the end. So with this type of sampling schedule, we're able to understand the role of lights in the attraction or avoidance by various species. So what the effect of the lights. This is a schematic of our, maybe I should turn off my email. You guys can hear this noisy things? Let me quit my email here. Okay, so this is a schematic of our data analysis pipeline. So essentially, if you see the stacks there of videos, sonar, and hydrophone data, which we have synchronized, of course, we are moving now from a stage of manual annotations where we were able to identify species and identify important behaviors that we wanted to measure or study in more depth. Some fish vocalizations, study the background. So we are at the stage of developing training data sets, and we're starting to move towards some machine learning applications. And of course, at the last stage, we want to be able to understand the fauna responses to environmental cycles, diel, tidal, seasonal cycles, and those introduced disturbances and the effects of artificial lights and organic enrichment, which we use bait in many of these experiments. And also in terms of the fish vocalizations, what controls those? Those are some of the basic questions we want to, at the end, be able to answer. Here's a graphic that was put together by Xavier. This is based on our, we have a automatic detector of fish or invertebrates based on the ARIS sonar. So you can see here, those are the 2 deployments we had in 2017 in the Strait of Georgia. You can see that the first deployment, you can see that the y-axis is the daily count of detections. The signal of the sonar essentially quickly degraded because we are at the mouth of the Fraser. It was a huge dump of sediments on top of the sonar head, so we lost the signal pretty quickly. But the second deployment in November 2017, we had a pretty good data set formed, at least a few months, two and a half months of good data, which we based most of our analysis. So what this is like the graph that you saw on the earlier slide was a post-processing data product, essentially. So it's a MATLAB script that was developed with help of Xavier, myself, and some of our data analysts at ONC. So essentially, you are able to actually download not only the videos and the sonar data, but you also can have this different data product, which is the sonar, the movement detections. And this is, if you click on this link, you can describe what the product is, what the data product consists of. Essentially, we set up a threshold of the backscatter return of the sonar, and the signal had to be consistent over a few frames. And of course, we delimit a threshold of size, and then we then classify as a detection of any organism in the field of view of the sonar. So here, some results of based on the video and the comparison between the ARIS sonar and the video. Of course, and expected for this such a highly turbid waters of the Fraser, the acoustic camera is way more efficient in detecting animals. You can see in red, the red bars against the black bars of the video. And this is standardized by hourly detections and also by the field of view of each of the systems, which are not exactly the same. So we have a 5-meter range, for example, here for the sonar, and the camera sees a little less because of all this turbid environment. So, and this graph was put together by our student, Madeleine Thompson at UVic. But of course, sorry, I forgot to mention, but of course, I'm not presenting here the results on the taxonomical identifications, but of course, with the video camera, when the turbidity signals are not too bad, then we can actually identify species. And we haven't moved towards identifying species yet with the sonar, but that's something we want to be able to do using machine learning as well. Here are some preliminary results of the effects of lights on and off. Essentially, on the graph on the left, you see that the detections are substantially statistically significant higher when the lights are off compared to when the lights are on. So essentially, the animals are avoiding the lights. But we had this interesting result from this from Hydrolagos, Collier, and Chimera, where we found actually this fish actually hangs out much more in the field of view when the lights are on. So essentially, you see in the right graphic there, after elapsed minutes from 6 to 11 minutes of lights on, you can see more the Hydrolagos hanging out in the field of view. So it could be an indication that it's attracted to the lights, but it's kind of not a firm response, because there could be other factors as well. But we're still kind of further investigating this. And here on the second iteration of this experiment, we deployed it in Barkley Canyon node, one of the sites where Jasper mentioned we have some of the hydrophone arrays. Essentially, it was pretty similar to setup, although we mounted the sonar below the camera, which optimized a little bit the field of view overlap. We had the hydrophone pretty well in view of the camera, and we added this bait-plume dispenser. So essentially, this is a modified carousel of a sediment trap, and we synchronized the release of each bait plume. Essentially, each bottle has scraps of salmon or halibut in cooking oil, and we synchronized the release of bait every 4.5 days, and with the video. So we saw the bait odor plume release in the videos, and we're able to see the response of the animals. So essentially, on top of the research questions,
1 minute here, Fabio.
Okay, so on top of the research questions we had in the other experiment, we now are able to kind of see the effects of the scavenger attraction and ecological succession after the introduction of this organic carrion. So this is the location we deployed. This is some preliminary results where we see a visually, this is all manually annotated data. So here, no machine learning yet, but we were able to see visually how the community changed over time. And there was a weak response related to the introduction of bait. Only the hagfish responded to the introduction of carrion. This is the third experiment that we just deployed a few months ago. And oh, I forgot to mention that the previous one, the acoustic camera didn't work. So, but now in May, we were able to deploy the Didson sonar combined. We had a modification of the bait system. Now the bait is actually released on the seafloor. We just essentially put upside down. And we have pretty good, it took us a while to set up the sonar, but it's now working perfectly. And we're gonna be able to do a lot of good analysis. Okay, finally moving on to 3 examples of machine learning. So we just published a paper based on video imagery from 3 locations of Barkley Canyon. We use a convolutional neural network, the YOLO version 5. We annotated some almost 3,000 video clips, almost 9,000 sablefish were annotated, and we found a pretty good detector with 97% of average precision. And essentially it was a very good algorithm even to detect the species under very different environmental conditions, lots of sediments in one site, and compared to the other. So this one is the Declan McIntosh is going to present next, so I'm not going to dwell too much, but we're essentially interested in startle response by sablefish. We're hypothesizing that the fish, they're just jumping and going, diving into the sediments, and we're trying to understand if the light is affecting that or is related to predation events. Anyway, I'll let Declan describe this project on machine learning, where we automated the detection of this startle responses. And finally, this is one project that is very enigmatic. We have lots of folks
Fabio, if we can move on to the summary, we might actually have to move on to Declan's.
Okay.
Sorry.
That's my summary slide. Next steps, we want to continue to partner with computer scientists to accelerate data analysis and interpretation of various projects. We want to support more students, undergrads, masters, and PhDs to use ONC data in their research. We have now launched the ONC's new Roy Heinemann Ocean Observing Award. So 2 students per year can apply and get granted $20K towards their graduation studies. We are developing in-house and with the help of collaborators some tools for simultaneously processing multiple data streams, video, audio, and acoustic imaging as a multimedia display, where you can quickly see the signals from all the data streams. We want to continue to explore the detection automation of specific behaviors. And we just had the interesting new visitors to the Fish Experiment in Barkley Canyon. And we have quite a few sightings of elephant seals, and we wanted to explore this behavior. We're now here at 650 meters depth. So we want to, we have colleagues working on this event. Thank you. Sorry it took a little longer.